Very good. So I will try to be brief in my presentation. There has been a small change in the order of the slides and small changes. So just because of the short period of time, I will try to basically focus on our major program, the most advanced, that is for idiopathic pulmonary fibrosis. What I wanted to tell you is that basically as that program forms the basis for where the company now is starting to focus what is in fibrosis. So to advance. Oh, thank you. So, well, the typical disclaimers, sorry. But let's go directly to our IPF program. We have a novel target for the treatment of IPF. And this is different than what everybody is doing, going after TGF-beta or TGF-beta pathway. This is a completely novel target that it came out from an analysis of single-cell RNA sequence. The protein is the actin-related protein complex 2. We have in a small molecule that is an inhibitor or modulator, however you want to characterize it in that language, but it has demonstrated antifibrotic activity in vitro using human lung fibroblast cell lines as well as in human precision-cut lung slides and in vivo in rat and mouse bleomycin fibrosis model. Again, I'm not going to go into IPF and the major unmet medical need that is there, basically only to say that there is 2 drugs that have been approved, and however, sort of the efficacy or basically the clinical benefit from those drugs is a kind of limited, reflected in part because basically 50% of the patients elect, even with this devastating disease, elect to not continue therapy. I told you that basically we have a novel target. The way that we identified it was basically taking a completely novel approach, looking at single-cell RNA sequence from a cohort of IPF patients, and we focus on so basically looking at the normal lung fibroblast and how those normal non-pathological fibroblasts become pathological fibroblast or myofibroblast. Doing that analysis, we identify ARPC2 as potential target in there. We validated using shRNA. So when we basically, sort of, I point you to the panel C, when we done, what happened? Okay, I have difficult finding so the pointer. So when we down-modulate the expression of ARPC2, you can see that basically cells, even when they get treated with TGF-beta, you have basically a reduction in fibrotic markers, both, in this case, fibronectin and alpha SMAD. ARPC2 is part of a complex, or it has been in the literature described as part of a complex that is known as the actin-related protein 2,3 complex. There is very little known in the literature about this complex, except for the fact that it is involved in actin polymerization, and more particularly sort of in the branching of actin polymerization. What I want to point out to you is sort of don't think so much on ARPC2 as targeting actin polymerization. We have data that basically speaks to the fact that ARPC2, while it is a part of the complex, it has a unique function. And we know basically on our molecular studies, basically blocking the expression of the different members of the complex, that only when you block the expression of ARPC2, you see an antifibrotic activity. You can block the expression of the other members of the complex, and there is no antifibrotic activity seen there. Just basically to show you sort of where, or to give you an indication of the target and where basically we have focused our medicinal chemistry effort based on the structure of ARPC2. That's at the point where we basically are going with our small molecules. A little bit about the mechanism of action. Again, when we first identified ARPC2 as a target, was completely being agnostic in terms of the biology of the target. And we have for a while a hard time to basically trying to understand why protein that originally was just basically implicated in actin polymerization will have an effect on fibrosis. We found out that actually and again, that the transcription factor MRTF-A that is well characterized in the literature as a transcription factor responsible for the production of for the fibrotic response and the production of fibronectin, collagen, and many other proteins involved in fibrosis. It's affected by the expression of ARPC2. And ARPC2 is required for the transfer of the cytoplasmic MRTF from the cytoplasm to the nucleus. Under normal conditions, MRTF is located in the cytoplasm bound to monomers of actin. When the cells get activated by TGF-beta or stress fibers or other factors, Rho kinase is activated. That activation of Rho kinase induces a dissociation of MRTF from the monomers of actin and allowing the translocation of MRTF from the cytoplasm to the nucleus. When we basically, and I point you now to the lower panel, when we take MRC5 cells and we treat it, and maybe it's even easier just if you go to the histogram, looking just basically at nuclear localization of MRTF. When cells get activated by TGF-beta, you see an increase in the nuclear localization of MRTF. Cells that express, where ARPC2 has been down modulated using a small interfering RNA, specifically target ARPC2, you see a down modulation. I'm sorry, my eyes are not working well and don't see the pointer. So when ARPC2 is down modulated, you see a reduction in the levels of ARPC2 in the nucleus. When you down modulate ARPC4 or ACTR2, two other members of this complex, you don't see an inhibition of the translocation. So MRTF is capable of going from the cytoplasm into the nucleus. Going more into this mechanism of action and just actually following what has been described in the literature, as I mentioned to you, that the translocation of MRTF from the cytoplasm to the nucleus is depending on the release from actin. We look at MRTF and actin interactions using the proximity ligand assay. So when MRTF and actin are together, you see fluorescence. When there is a dissociation of MRTF from actin, the fluorescence will be gone. Again, the figures are difficult to see. I point you to basically at the histograms, that is basically the quantification of those phenomena, similar to the previous experiment. Now looking basically at control cells, this time using a small interfering RNA targeting ARPC2. Again, control cells not stimulated. You see a high level of fluorescence indicative of the interaction between MRTF and actin. When you treated the cells with TGF-beta, there is a decrease in the fluorescence indicative of the dissociation of the complex of MRTF and actin. When you basically, again, remove ARPC2 in control cells, nothing happened. That's surprising in there. But now when you stimulate the cells with TGF-beta, you see that that decrease in the fluorescence is not happening as it happened in the cells that express normal levels of ARPC2, showing that basically ARPC2 is required for that dissociation induced by TGF-beta. The other way to look at that, I point you now to panel C, when we basically now what happened when we overexpress ARPC2. So instead of reducing the levels, now increasing the levels. Similar experiment, now again looking at just basically cells that have been transfected with an empty vector. You see good levels of fluorescence, again indicative of MRTF and actin monomers being together. When cells get transfected with different levels of the vector, again, reflected by those 50, 100, and 200 MOIs. You see that basically now overexpression of ARPC2, you don't even need to treat the cells stimulate the cells with TGF-beta. There is a dissociation induced just by the presence of overexpressed ARPC2. Again, I think those basically are two nice complemented set of data. So this is our current hypothesis that we are trying to define even in more detail and looking at other potential arms of the mechanism of ARPC2. So under our current hypothesis, MRTF is bound to G-actin under normal conditions. When cells get stimulated and receive a fibrotic signal, something happened to ARPC2 in a way that now ARPC2 will interact with the complex of MRTF and G-actin, and probably sort of just competing in that binding and releasing G-actin from the complex, allowing MRTF to go to the nucleus. Our compounds, by binding to ARPC2, block that interaction and sort of just keep MRTF in the cytoplasm, render it inactive. So let me now switch gears a little bit and just basically showing some of the data that we have with our compounds. I will point out that we have also the type of studies that I showed you before in terms of nuclear translocation of MRTF, as well as the interaction of MRTF and actin has been initially reproduced with our compounds, but we need to basically do more experiments to be confident of that data. So, but just now looking at basically a fibrotic response induced by TGF-beta in MRC5 cells. Again, the way that the experiments are done, we see the cells on day 0, on day 1 we treat it with TGF-beta and our compounds, and basically 4 days later we just measure the levels of fibrotic markers. In this case, an example, alpha SMAD. Of course, you see a sort of untreated cells, very low levels, treated with TGF-beta, increase in the levels of alpha SMAD, treated with the compounds, a reduction or inhibition of alpha SMAD production. And then on the right-hand side, you basically see a sort of the IC50 for these two compounds. Besides looking at normal human fibroblast, we have also looked at human precision-cut lung slides. These are human tissues, human normal tissues that can be kept in culture for more than a week, and just basically represent a sort of a complete structure of the normal tissue with all of the cellular interactions that it will happen in vivo. So trying to resemble it, what it will be as sort of a closer as we can to the human conditions. In this case, again, the tissues are put in culture on day 0. On day 1, they get stimulated. This time, instead of basically using only TGF-beta, we actually use a cocktail of profibrotic agents. TGF-beta, PDGF, IL-11, and a whole bunch of other things. So the cells get stimulated, the fibrotic process gets initiated, and then 48 hours later, we initiate the treatment with our compounds. And then at day 6, we just basically harvest and analyze the levels of expression of different fibrotic markers. Again, as an example of some of the fibrotic markers that we have looked at, you have here collagen type 1, ACTA2, CTGF, fibronectin, and vimentin. We have analyzed other additional ones, but the picture is pretty much sort of the same across the board for all of this. Different doses, different response. I give it to you, sort of there is variability in the assay. There is variability also from sample to sample. This is just basically represent the study from one donor. We have done repeated up to now 6 donors. The picture is always the same. Our compounds basically sort of reduce across the board all of the fibrotic markers that we have looked at. We use nintedanib as a positive control drug that is approved, so we expect that we'll see again some effect in there. We see some effect what I would like to point to you is that the profile is different than of our compounds, that the profile that you can get out of nintedanib. That's as far as I think I wanted to take it at this point. Different mechanisms of actions reflected on a different profile in terms of inhibition of fibrotic markers. Efficacy in vivo, the mouse bleomycin model, again using as a therapeutic model, induction of lung fibrosis by tracheal installation of bleomycin on day 0, and then on day 7 starting the treatment of these animals. On day 21, just basically harvesting the lungs, do histochemistry and analyze for fibrosis on those, and basically score the level of fibrosis using the Ashcroft score system. And panel B, you basically see a sort of vehicle-treated high levels of fibrosis, treated with the compounds, again, 2 different compounds at 30 milligrams and 100 milligrams per kilo twice a day orally. Dosing, you see inhibition of the fibrotic scores, and you have nintedanib also sort of as a positive control inhibition of the fibrosis score. Our current clinical candidate, AI, and sorry for the mistake, should be 1640. Also sort of doing a dose response together with nintedanib. What I want you to take out of this is basically the 30 milligrams per kilogram per day total dose, but given BID is an effective dose. We have done the PK in both rat and mice, and what I will want to basically point out of all the PK data is the good oral bioavailability of these compounds. Again, the T1/2 is about 2 hours of those. That will explain the sort of that you actually have to give it twice a day. With twice a day, we are basically covering around the IC50 of the compound. We're trying to give it sort of once a day, and even if we double the dose, we don't get efficacy. We have basically going into the selectivity profile, looking at basically a panel of receptors and enzymes. Out of that analysis, when we basically use 100 micromolar as the highest concentration in those assays, only the 2 of the serotonin receptors come out as sort of as hits here. None of the others sort of have shown any effect at that high concentration. We have, again, with our lead candidate, and I point you to the bottom, completed non-GLP 14-day tox study, 30 milligrams per kilogram dose was completely clean. We see toxicity at 1,000 milligrams per kilogram. One of the things that we see is actually a reduction in the levels of lymphocytes. As well, basically, sort of a salivation, change in salivation. For a previous compound that we have also sort of now put it on the side because the selectivity profile is not as good as 1640. We have been able to go to a 4-week non-GLP study, and the picture is, as you see, is the same. It's sort of nothing up to 30 milligrams per kilogram 30 milligrams per kilogram per day. So just basically, let me bring you back to, so the effective dose in the in vivo models was 30 milligrams per kilogram per day. With the 14 days study, 300 milligrams per kilogram per day, it was safe, no toxicity. So sure, as we move forward, we'll probably sort of that safety margin will get reduced, but at this point we have a 10-fold safety margin, what I think is pretty reasonable to move the compound forward. So as a bottom line, novel target for the treatment of IPF, a small molecule that has composition of matter, protection in terms of the composition of matter, novel mechanism of action, potential to be a first-in-class, and again, as I mentioned, sort of efficacy showing in vitro in vivo in fibrotic models, and we are now basically sort of initiated IND-enabling studies with expectations that these studies will be completed around the sort of July of next year, aiming basically we have a partner to move with us or funding move into the clinic for first in human studies towards the end of next year. And I think basically just wanted to conclude it with there and open up for any questions, anything that